Klebsiella pneumonia. It's a gram-negative rod-shaped bacillus bacterium. It's a facultative anaerobic, non-motile, and has a prominent polysaccharide capsule, which plays crucial role in virulence and evasion of host immune system. Klebsiella pneumonia is an opportunistic pathogen that can cause pneumonia, infection in urinary tract and lower biliary tract, infection in a surgical wound site, as well as bloodstream associated infection. In the most vulnerable patient are neonates and the elderly, especially those that are immunocompromised. The prevalence of Klebsiella pneumonia infection vary globally due to factors like geographically and depends on factors such as healthcare infrastructure, antibiotic usage, and infection control. Klebsiella pneumonia strain often resistant to multiple antibiotics, including extended spectrum beta-lactamase. In antibiotics like third generation cephalosporin, treatment option may include carbapenems, aminoglycoside, and fluoroquinolones. Okay, virulence factor of Klebsiella pneumonia. It's a Klebsiella pneumonia have a thick capsule polysaccharide layer shield it from the immune system, preventing immune cells from clearing the bacterium. This help Klebsiella evade host defenses and establish respiratory tract infection. And release harmful substance toxin that damage tissue and cause inflammation. Then the inflammatory response causes the body to release a pro-inflammatory substance like interleukin 1 and tumor necrosis. Too much inflammation can harm tissue and make pneumonia symptoms worse. And then the antibiotic resistance make treating pneumonia harder. And lastly, is biofilm formation is a form of slimy layer called a biofilm on the surface on the respiratory tract or on medical devices. So it can stick, Klebsiella pneumonia will stick together with the form of slimy layer. Okay. Problem statement, difficult to recognize or understand this Klebsiella species make the treatment process delay. So most of the bacteria Klebsiella identified currently is multi-drug resistant, and it makes challenging to treat the infection caused by these strains. Several factors contribute to the development and spread of multi-drug resistant Klebsiella pneumonia, like overuse and misuse antibiotic, healthcare setting, international travel, and lack of new antibiotic. And the third one, there is a lack of comprehensive information on bacteriophages, secondary metabolites, and genomic island in Klebsiella pneumonia that could contribute to its pathogenicity. Research hypothesis. By identifying and characterizing genomic island, bacteriophages, and secondary metabolites in Klebsiella pneumonia, the study could provide new insight into the mechanism of pathogenicity in this bacterium. The genomic element identified in this study could potentially serve as target for the development of new therapeutic strategies against Klebsiella pneumonia infection. By comparing local strain of Klebsiella pneumonia with database strain, the study could contribute valuable data to the field of comparative genomics and enhance our understanding of the genetic diversity and evolution of the Klebsiella pneumonia. And the finding of this study could pave the way for future research, such as functional studies of the identified genomic elements or the development of novel therapeutic approaches. So objective of my research is to perform comparative genomic analysis of 30 pathogenic Klebsiella pneumonia genomes. And the second objective is to investigate the evolutionary relationship among 30 pathogenic strain of Klebsiella pneumonia through phylogenetic analysis. And the third objective is to characterize the genomic landscape of pathogenic Klebsiella pneumonia strain by identifying and analyzing genomic island bacteriophages and secondary metabolites. Briefly, my methodology is firstly need to genome assembly. Total of 25 complete sequence Klebsiella pneumonia bacteria were chosen from NCBI database and 5 sample from HUSM. And then the sequences of 30 genomes were retrieved in a PASTA format file and well known for its pathogenicity. Genome annotation RAS to identify the presence and type of pathogenicity factor present, seed subsystem used to screen pathogenicity and virulence genes. Okay, and then next phylogenetic analysis using MegaX software, 16S RNA sequences retrieved from RAS aligned by the cluster W and neighbor joining tree. The screening of secondary metabolite on web server anti-smash was used to screen the secondary metabolites produced by the 30 genomes. And screening of bacteriopages, PESTER web server was used to identify propage sequences within the selected 30 genomes. Okay, as my first objective is to perform comparative genome analysis in 30 pathogenic Klebsiella pneumonia. Firstly, I collected 16 sRNA sequences with our 99% similarity in their sequence to our local 5 strain based on data assessed on March 15, 2023. Okay, the sequences of the 30 genomes were retrieved in FASTA format file and well known for its pathogenicity, and the source of sample from pairings, wound swab, intra-abdominal abscess, burn wound, rectal swab, miscellaneous body fluid, and for our 5 local strain, one sample from blood and 2 sample from trachea aspiration and 1 sample from sputum and another 1 sample is from urine. Okay. And then for 5 local genome strain, no GenBank accession number because haven't deposited the strain in database. And then after this, after compile and save the data file according to the genome strain, then done annotation for all the 30 genomes with rapid annotation system technologies RAS tool. The annotation process for each strain respectively take between 3 hours to 1 day. This is the data that I extract from the subsystem seed for the 30 genomes. And then to further facilitate comparative analysis seed substance by RAS server, the seed subsystem is a platform that supports various comparative genome analysis and it's used across thousands of genomes to provide consistent and accurate genome annotation. Based on the seed subsystem, the largest functional group for all the species in amino acid derivatives count around 263 to 284, followed by the carbohydrate from 398 to 401, and membrane transport, and moreover functional gene cluster under protein metabolism. Functional group consistent of cofactor vitamins. Prosthetic groups also count for most of the CDS count across all the 30 Klebsiella pneumoniae strain. Between the range of 156 to 187, and the analysis and the prediction of the functional gene will help us conclude the type of subsystem category available within the Klebsiella pneumonia. In this all data, I just particularly focus on the functional group named virulence, defense, and disease. It's why I choose this subsystem, because virulence factors are mechanism processed by Klebsiella pneumonia pathogen and enable them to cause disease in the host. And this functional group is subdivided into several other groups. These groups include adenosine toxin and super antigen, bacteriocin, resistant to antibiotic and toxin compound, and lastly invasion and intracellular resistance. Among the subdivided group, only two groups, namely antibiotic and toxic compound and invasion and intracellular resistance, were recorded to have a gene cluster present by the seed subsystem. So the first subdivided group resistant to antibiotic and toxic compound contain four main subcategory, like cobalt, zinc, cadmium resistant, copper homeostasis, copper tolerance, zinc resistant, and copper homeostasis, and lastly beta lactamase. And then invasion in intracellular resistance. Invasion means Klebsiella pneumonia can invade tissue and cell using structure like fibrae and adenosines. This help it to attach to the enter host cell, causing infection. Once inside, Klebsiella avoid detection by the immune system and multiples, spreading the infection. So, and from here, the Mycobacterium virulence operant possibly involved in coloninated is a key intermediate in the biosynthesis of nicotinamide adenine dinucleotide NAD. As essential cofactor involved in various cellular process. Virulence operants in bacteria typically consist the gene that encode protein involved in various aspects of virulence, including adhesive invasion, evasion, and of the host defenses and modulation of host immune responses. These operants often play crucial roles in the pathogenesis of bacterial infection. Then for the Mycobacterium virulence operant involved in DNA, there's no direct evidence of the specific Mycobacterium virulence operant influencing Klebsiella pneumonia pathogenicity through the DNA. And my next objective to investigate the evolutionary relationship among the 30 strain of Klebsiella through phylogenetic analysis with the Vibrochlora as an outgroup as was performed. Uh, was performed using molecular evolutionary genetic analysis MEGA-X software. 16S RNA sequence retrieved from RAS annotation file were aligned by the cluster W and construct neighbor joining tree with bootstrap 1000 was construct in MEGA-X software. And then based on the phylogenetic tree, it can be seen that the selected Klebsiella pneumonia in group into distinct cluster, cluster A and cluster B, and strain of IRQBAS 103C, and then strain 0113481141 and strain INF 298. Each represent a distinct cluster and are distantly related to other species. And outgroup with Vibrochlora is used as a point of comparison for in-group genome strain, especially allow for the phylogeny to be rooted. So my objective 3 to characterize the genomic landscape of pathogenic Klebsiella pneumonia strain by identifying and analyzing genomic island, bacteriophages, and secondary metabolites. The updated version of anti-smash web server was used to screen for presence of secondary metabolite gene cluster within the selected 30 Klebsiella genomes. Microbial secondary metabolites are small organic molecules that play crucial roles in the microbial cell culture survival and ecologically interaction with other organisms. They are involved in cellular activities such as signaling, regulation, nutrient scavenging, and self-preservation. Secondary metabolites produced by any bacteria, fungi, or plants are of great importance of human as they are manipulated for the benefit of humankind. So here we can see the enterobactin substance is almost in all the 30 strain found in all the 30 strain that helps it grab iron from the host. By grabbing iron, enterobactin helps Klebsiella grow and spread in the host, making it more harmful. And then the next one is the O antigen, also find the 29 strain of the Klebsiella. It's the part of the outer layer of the bacterium. It makes up of a sugar units that repeat and help form the structure of the bacterial cell wall. And the O antigen also shield the bacterium from stress and attack by the immune system. And Lancashidin C is a compound produced by the certain strain of Streptomycin bacteria, has been studied for the potential pharmaceutical properties, particularly as an antibiotic. However, there is no information available regarding its production or involvement in Klebsiella pneumonia. Comparative genome analysis using PASTEUR bacteriophages present within the Klebsiella pneumoniae genomes were identified using page search tool enhanced release Bester, as the presence of abundance of pages could provide insight into the pathogenicity and horizontal gene transfer even between Klebsiella pneumoniae species. And then the Pasteur result was used to rapidly identify and annotate page sequences within the 30 Klebsiella bacteria genome. The online pages finding tools identify the categories pages according to calculate score. The pages sign between the different color to identify the completes of each page sequence. All the 30 strains contain pages in chromosome, and for plasmid only this strain IRQBAS1003C don't have pages on the plasmid. Other than that, all 29 have pages in the plasmid and chromosome. And then next is a metabolite pathogenicity pathway mechanism using encyclopedia of genomes, genes and genomes KEGG is in progress, and genomic island discovery also is in progress. So this is my Gantt chart and my references. Thank you.
Okay, thank you, Yogeshwari. Eugene?
Yes, thank you, Yogeshwari. So we can see that you've done a lot of the bioinformatics analysis. However, I think in terms of the presentation of the information, it's a little bit too much because of the details that you shared. So in the future, I guess you can summarize more.
Okay, doctor.
Yes. Can you show your overall slides?
Overall slide.
Yeah, okay. So this is fine. Yes. So from all the different analyses, because you have phylogenetic and you have secondary metabolites and phages, can you summarize the connection between all of them?
Connection between all of them.
Yes, because you showed us like individual data or results, right? You have all the different analysis. So how can you tie all of them together?
Uh, For time being, I just tie with the virulence factor of Klebsiella pneumonia, doctor. And I didn't do further similarity of this all the soft result, because I haven't finished some more 2 more steps to go.
Okay, so in that case, can you maybe share what do you think will be the most interesting result or relevant result that you have uncovered from your analysis?
The pages, doctor, because all the Klebsiella strain that what we analysis all have pages which it shows that got what horizontal gene transfer, doctor, between the Klebsiella.
Okay, and in the phylogenetic tree, we can see towards the end there is one group or cluster which is similar or very near to Vibrio?
Yeah, doctor.
Yes. So did you include Vibrio as a control only, or is it because Klebsiella pneumonia is also very closely related to Vibrio cholera?
It's because Klebsiella pneumonia is very closely related to Vibrio cholera, doctor. It's one of the sister group, right, doctor?
Okay. So can you show me your last slide?
This one, doctor?
Maybe the one before that. Yeah. So you have two more to go, and so usually in a progress presentation, we need to also provide a sort of summary. I think we can see all your data, but it will be good to summarize. Therefore, since your work is still in progress, it's a bit difficult to comment now, but I think we can say that you've done a lot of the analysis and work already. So I hope in the next presentation, you can tie all of them together, because you are just showing the individual results, but we cannot fully understand what it all means.
Okay, doctor. So need to summarize with all the result that I get, right, doctor?
Yes, so you need to summarize all of the results and then connect all of them together, because you're giving a global genomic analysis, right, on all these strains.
Okay, doctor.
Okay, that's all from me. Thank you.
Thank you so much, doctor.
Thank you. I think that concludes the session for today.
Okay, thank you, doctor.
Thank you. Bob, itu saja kan?
Itu saja, cuma nanti examiner kena isi borang rubrik tu lah, dan email saya dengan
Itu lepas kelas lah, nanti petang Dr. Eugene boleh buat lah.
Okay.
Alright, thank you everyone. See you.
Okay, thank
Bye.
you.
Okay.
You should be able to log in with your student ID. This is mine, right? I logged in already, and you can download the USM thesis here. Example, yeah, if I search my name, then my thesis from 2011 is here. And more recent ones, perhaps, if you want to search for ELISA. ELISA. And these are all the articles about ELISA that you can download. So these are from master's level and sometimes undergraduate level as well. I think mostly a master's level. So you can see all the previous students' ELISA. So for example, this one is from 2020. It's from Science Perubatan, which is the medical school of medical sciences. And download the thesis, of course. So this is a Master of Science, Biomedicine Mixed Mode, right? So you can see the examples of the thesis for almost all. They don't upload all of them. I don't know, for whatever reason. Sometimes there's a backlog or whatever. But most of the thesis you can download from this closed repository. So these are the full thesis you want to download as examples for your dissertation. So there are no page limits to the dissertation. Usually, I think after you have included all your results and write-up, at least it will be about 50 pages, right? So usually it's about 50 pages. And if you write very compactly, 50 pages is okay, it's acceptable. So there's no limit. Doesn't mean that short is bad or more pages is good. Okay. So we can see from today's two examples of presentation, there are different presentation styles and also different way of preparing the slides and even different ways of speaking or presenting your results. So which one do you think you can understand better? Which one do you think you can understand the project better? First one. Okay, so why do you feel that way?
I think that
Okay, my mic is on. Yeah.
really expect I think
Yes.
it's
Mm. Yeah. So
second one
mm,
is
yes. So it is up to you how you want to control your own presentation, like how much you want to present or how little you want to present. So more than 20 years ago, when I was an undergraduate at university in Sarawak, just like Leo, so I had this professor who said something which will sound very sexist today. So he said, so this is a Muslim professor, so he said, presentation or scientific communication should be like a mini skirt. Why? Why is it's like a miniskirt? So he said it should be long enough to cover the essentials, but it must be short enough to be attractive. Do you understand what is meant by that? So today, if you say that, then you'll get reports from the student, and somebody will complain to the management or the director, right? If you say something like this, yeah. Yeah, but that was 20 years ago. So I think what he's saying in a good way and an analogy that is suitable for that time period is that our presentations need to be long enough so that it covers all the important information, right? But not too long to the point where people start to uh, think about something else, which means that they lose their focus, right? So the reason why we need to control the information. So I'm going to close this. Then you can download this later on your own. Okay. So the reason why it's really important to control the information is because, like I said just now, our world is a distracted world where we live in. Okay, so I'm going to show you an example of slide that looks like this. So this is an actual slide. Okay, so previously I had a student from Nigeria. Then he participated in a conference in Nigeria. And this is the slides. Okay, so it looks like this. Then this is the introduction. So the good thing about this slide is that the conference sets a limit that there must only be 7 slides and you have 7 minutes to speech. Because at that time it was during the pandemic, so it was an online conference, so you just record your voice over this. Okay. So this is what the slides look like. Essentially, the result is about antibiotic resistant genes. This is the, I close this back. Okay, and these are the references. All right. So after he showed me this, then I helped him to change the slides. Okay, so this is the same information. So these are the same slides that you saw just now. Okay, but if we can rearrange the information or present it in a different way, then our slides would look easier to follow. Okay. So we have the title slide like this. Yeah. Okay. So this is the original one. So everything is the same. So all I did was to change the layout. Genetic relatedness in extended thing, presenting author and co-authors. So you can see the difference, right? In just changing the layout and changing the text. So during presentations, it is critical to think about all these things. However, it's not possible for me to show you everything in a short period of time. So later I will share these videos with you, which you can watch on your own time. Okay, so there is a playlist of videos. Mainly it is about graphic design, but because during our presentation, preparing our slides is essentially about following the rules of graphic design. So graphic design doesn't mean that we are going to make a pretty slide, but it is how we present our information so that it becomes clear, right? And that is how, whenever we look at magazines, they always look simple to us. Okay, so these are the original slide and the after slides side by side. So instead of presenting the introduction, antibiotic, antimicrobial resistance, if you can show it in a more graphical way, then it will be easier to understand. So that is why if you look at the two types of presentation just now, the first one has more simpler graphics. Instead of text, what you can do is to hide the text. Of course, maybe you may feel that this takes up a little bit more time, right? However, if you are able to do it more and more, then it will become easier. And there are several tools in PowerPoint that you can use to make it easier to edit, which I'll show you later. So instead of text like this, which is firstly very hard to see, okay? And secondly, if you have a flow chart, it is best to number them, because what you want is to indicate the sequence of your steps, right? So instead of an arrow, which we typically see like this, you can just number them so that it's straightforward. So when we present, we need to be direct in our communication. So the speaker has to do the work and not rely on the listener or the audience to try to read the speaker's mind. You know what I mean? Okay, so you have to prepare everything in the sense that everything is ready to be absorbed. Because when we present, just like what we saw in the earlier presentations about the I am only seeing those slides for the first time today. Only seeing for. And we only have maybe 1 minute or less than 1 minute to look at each slide. So there's always this psychological barrier between the presenter and the audience. We have, as a presenter, while we're preparing for slide, you already seen it multiple times, right? You are the one preparing the slide. You are the one that is doing the project in the lab. So you know the work inside out. You know the work very well. However, we also have this conception that the audience also knows the work at the same level as we do. So this is a kind of blind spot where it is normal because whenever we prepare, we are all by ourselves, right, working on our laptops, and we never really get to practice in front of somebody that has not seen the slides before, or even what our speech is before. So you need to make everything as straightforward as possible with as little distraction as possible. Even if you are going to show a table, you need to highlight what is the terms of the table. So there are 3 points when we need to figure out when we want to present our data. Okay. So the first point is what is the data? Of course, this is what we see, right? And then we need to tell the audience what this means. Okay, so what does this mean? So that is the interpretation of the data, because the audience cannot understand. They are not working perhaps in the same research area as you. Some of you may be working on cancer research, or some are working on natural products, or plant extracts, or PCR, and so on. So everybody is working on different projects. Therefore, something that is very basic, such as real-time PCR, could mean differently to the person who is working on the project than the audience. Okay, so we have to bear this in mind that the audience is composed of different people with different levels of understanding. So even if they cannot understand fully what is shown here, which is reasonable, right? Because we have like 1 or 2 minutes to look at this, but at least we can remember that, okay, maybe this is the most important result. Okay, we will not be able to absorb all this, especially with the all these initials and so on, right? So it doesn't make sense. So this is not a good way to present this data. However, the audience is not supposed to know everything. What they need to know is just what is the main point you would like them to know. Okay. And summary, okay, it's the same thing. We can conclude that, okay, what are the main findings? Even just by rearranging and adding color, and highlight what is important. So all of this comes from the knowledge of a little bit of graphic design, because graphic design is about how they present information, especially in advertisements, so in posters, in TV commercials, and so on. That is where they are able to convince the target customers or consumers to buy a certain brand of product and so on. So there are all sorts of visual things that are in the subconscious, meaning that if we are going to be more interested in something, then there must be something that is attractive to us, right? So if somebody's presentation is not interesting in the first few slides, usually you are going to stop listening to it. Okay, so just by this, there are so many information that you are already confused as to what the project is about. Okay, so I will share this afterwards. Let's see. Okay. Okay, so this is a talk that I give some time ago to the undergraduate, sorry, to the postgrads here in Nepal. 2 years before this. So a postgraduate is like a maze. So if you make your way out, maybe you finish your master's here, then you can continue on to another set of maze, which is your PhD. So of course, if you are doing a mixed mode, the challenges are different, but still in doing research, there are some unknowns, right? So that is what makes research interesting. Okay, so everybody sort of looks out of focus for a little bit. So doing science is a continuous process, right? So this is my slide from 1999, because this is the PowerPoint technology at the time. However, in after my PhD, so during my PhD presentation, my slide has sort of evolved to something like this. Okay, then I can also improve myself to present much more clearer. And what gave me the idea was that during I was doing my master's, so I applied for a grant. So this grant is called the Malaysia Torres Science Foundation. So during the year 2002, although we already have PowerPoint, still during that grant interview, we had to use transparencies. So nowadays, nobody uses transparencies anymore. So transparencies is a projector, a light projector, where you place a transparent film on top of it, and then it projects a light on the screen. So that is maybe a very long time ago. And even in USM, I think they've already removed most of the projectors from the lecture halls. Yeah, maybe some secondary school, you may still have the light projector where the teacher can write on the transparent film. Okay, yeah. So this was my research proposal at the time. When I showed this, so the panel said, "Oh, this is what the proposal should be." Meaning that everybody else was presenting in a kind of text-based proposal. So I was surprised also when I heard that. Yeah, this is what wanted to be. So it means that the audience would prefer more something that is visual. So visual language is the same for everyone. Okay, so if I were to say the word apple, it can mean different things to different people, right? Apple somebody may visualize it as a red apple, maybe 99% of the human population will visualize it as a red apple. One person may think of a green apple. Or you may think of Apple as a product, an iPhone or a computer, or can also mean different things. So that is why if you are going to show something, it has to be direct. So this was the title of the talk, the art of preparation of attractive slide for scientific presentation workshop. So, okay, I didn't give title myself, but was given by the coordinator. Before we prepare our slides, what are the first things that we do? Some of us will select the template, right? You can choose different templates, and you can see that different templates have looks differently. Some people prefer something like this. These are all the default PowerPoint templates. Maybe something like this, or something like this, right? So which one do you think will be suitable or more easier to read? So it depends. All of us have different preferences. However, a more simple one may be better, right? It may be easier to read. If it is the text is too bold or too fat, then it will be very difficult to read as well. So keep this thing simple. Okay. So this is presented, just before the pandemic in February 2020. So what we want to do is present our scientific presentation with simple slides. Okay. So later on, I'll show you what the duck is all about. Okay, so this is a quote that is attributed to Einstein. Does everybody know who is Albert Einstein? Yes. Okay. And the quote can be simplified like this. Everything should be made as simple as possible, but not simpler. Okay. And here he is saying this. So a long time ago, as you know, everybody was writing in chalks on the chalkboard, right? But today, somehow with PowerPoints, everybody likes to, decorate their slides with non-relevant things, like some kind of background or something. But what you want is your data, and you do not want to see what the background is, because that will distract them, especially if your data could be important data, but if it is too many components, then by adding something else, if you do not understand this, automatically your brain will look for something that it can distract, right? Your brain will be attracted to something that is more comfortable or more safer to look at instead of looking at all these formula. Okay, so if there is no formula, no decoration, then all of us will just look at this, right? So unfortunately, we are in the age where we have access to too much information. So why do we present? Yeah. So if a tree falls in a forest and no one is around to hear it, does it make a sound? Huh? Have you heard of this saying before? If a tree falls in a forest and no one is around to hear it, does it make a sound? So the equal saying is, if a scientist sciences in the lab and does not communicate it, does it count as science? No? Oh, yeah, and this is a selfie taken in 2010. So I was waiting for my Western blot to develop and I was bored. So I just put the phone camera there and wait and pose while waiting for my Western blot. So whatever we do in the lab or in research, it has to be communicated or else it's just to yourself and your supervisor, right? So because research is to benefit everybody else, therefore, it is important that we communicate as clearly as possible. So we think that communication happens as soon as we say something. However, the only communication that is true is once the listener or the audience understands what we are saying, right? So if you can talk and talk for half an hour, but after your presentation, nobody understands anything, that will also not be good as well. And this is another way to present this information. So which one do you like? Is it this way? So when communication actually happens is once the other person or the audience actually understands what we are talking about. Okay. So it really depends if you give a duck or don't give a duck. And what is a duck? So duck stands for something. Okay, so if a person can tell me what, so duck is an acronym for something. Okay, and later on, if you can tell me what each of it stands for, maybe you can get a prize from me. Understandable. Okay, it's good. So what does D mean? Yeah. Description. Okay, it's close. So this is about a scientific presentation. So these 4 letters represents what we need for a scientific presentation. Okay, so do not take up everybody's time. Okay, so D stands for data driven, which means that whatever we present, it has to be based on our data or data that has already been published before. So there is a precedence, meaning that it is not something that is made up entirely. It must be understandable because if no one understands the data, it is also not useful. Okay. So you have to simplify the data to make it understandable. Understandable is just meaning what we can comprehend or understand, right? But in order to help us understand, it must be clear as well. So there are no distractions from our data. We shouldn't decorate the slide so that it is, over decorated, too much fancy stuff that distracts. In the past, people like to use some like animations where the slides kind of move about. Luckily, these days nobody does that anymore, right? Like sometimes I think you remember many years ago there's also sound effects like you had explosions while the text is coming in. And finally, the data, our presentation must be kind because if it is not kind, then everybody will be bored and not listen to us. So kind means that we need to be kind to the audience. So the presenter must do all the work, must prepare and think about the audience. So it's not about being egoistic and presenting our data to say that we have done this work in the lab. I did not sleep for 3 days because I do the experiment. So it's no longer about what you did in the lab. So whatever you've done in the lab, not to say that it is not important, but when you present, yeah, it shouldn't be about what you have done in the lab. It's about what you would like to share with the audience for maybe 20 minutes today. Okay, you only have 20 minutes, but perhaps you've worked 20 months in the lab to run your PhD experiment. How do you simplify or share that experience of 20 months into 20 minutes? So there are many books that you can get from the library and also online, such as these books. Okay. This is also a poster that you can download online. Later on, I will share this with you as well. So these are just some basic guidelines that we can follow to help us present and prepare our slides better. So that will mean giving structure to our presentations. If you look at Stephanie, the first presenter just now, she also segmented her slides so that there is like the title and the information. So that will help us and the audience to know that what they should look at when they are listening to you talk. Because there are many things that are happening at the same time. You want to understand the slide, but at the same time, you are also listening to the presenter. So your brain is doing at least 2 things, right? It is looking at the screen, it is listening to the presenter, and at the same time, maybe a third thing is trying to analyze the data. So you shouldn't let the audience analyze the data. So you should prepare everything in advance before we do that. Okay. So here is also a very interesting or informative video that describes how we can structure our talk. And there are many, many more on the internet, on YouTube especially, if you would like to learn how to clean up and prepare your slides. BioRender has become useful these days. Okay, so BioRender has only been around since 2019 or 18. It is free. However, you cannot export, I think, in the high quality mode. Yeah, but it's still free, so it's good that you use for your internal presentations. You have not used this before? BioRender. Okay, so yeah, it's free to use inside, and you can export certain figures as long as you attribute, meaning that you say this figure is created by BioRender. So we have to figure out the data-driven, understandable, clear, and kind figures. So one example is like this. Okay, so this is my students, a master's student's draft figure. So actually she did a good job already. Okay, yeah, she did a good job already, but it can be simplified a little bit better, I think. Yeah, so it depends on how you want to make it clearer. So even like this, by making the font size a little bit bigger and bolder, and to get rid of the unnecessary element, like for example, if this line indicates here, then if you have a clear title, it will be understandable as well. So you don't need this line anymore. Because the more element, then the more confusing it becomes. So your job is to prepare in advance for your audience and what they need to do. This is another example. So now there are 3 elements in a slide. So this is also useful when you're preparing your manuscripts for publication in the future. There are 3 elements in the slide, in this 3 elements of data that you want to present. One is perhaps this information about a certain gene. Another one is your agarose gel picture. Another one is southern blot. This is a blot. And if we want to simplify it, we can simplify it like this. So this is the main information. Okay. So just like what I said, there are many steps that is going on here. Of course, all of them is important. But what does the audience need to know? Yeah. So if you put it like this, it's still the same information. Let Z, okay, here. Let Z and the cut F gene and this APH1. So this is the same slide. Instead of numbering it 1, 2, and 3. So all of us typically just copy and paste our results from our lab notebook, right? Or our data. So lane 1 and 2 and 3. So you need to tell them directly what is lane 1. WT in this case stands for wild type. Wild type, and this is the other variant. So they know they do not need to read the legend. 1 is here. So when we present, it is different from a journal article because a journal article we can take our time to read the journal article, and we can go back and forth between different pages, right? If you want to take 5 minutes to look at this figure, it's fine. But when we present, we only have maybe 1 minute on this slide. So we should tell them directly, okay, this is what size and this is what size. They should not have to do the analysis in their heads. What they should do is just look at the screen and listen to you present. Okay, so you need to add all the information so that it is clear to the audience straight away without them having to read the legend. Okay, here is an example of when we do a journal club. So in some labs, they also have this practice of doing journal clubs, right? Journal clubs is when we read an article and we share it with all our different lab members. So a journal club is just the same as a case study inside our mixed mode course. So in case studies, you also take an example from a journal article and present it. Okay, so if we have something like this is an example. Okay, so this is a journal club from my friend, and she used this. So as I mentioned, when you have a decorations like this and different colors and so on, it becomes distracting. And also, in this case, the gray and white doesn't match so well because it's difficult to read. Yeah, so just by removing the background and simplifying a little bit, then it can be slightly better. So here, you can also simplify the title so that it is directly understandable. Okay, you can simplify the introduction so that people know what this is all about. So you want to highlight, of course, this, right? And maybe you want to show that, okay, inside this bacteria, it can accumulate this red thing, polyhydroxyalkalides. So what would be better is maybe if you add a red arrow here, then that will be clearer as well. So another way is to present your information in a graphical way instead of introduction with bullet points. Because if you have bullet points, then, like I said just now, somebody reads the text. Bacteria accumulates CH. Everybody will envision it differently because you do not know what it looks like. So if you want something that to be known what it is, then you have to show them straight away. Okay, so these are all the details. So you need to go step by step in certain cases. So in this case, this information is very complex, right? However, when you present, you need to layer your information. Layer means that you do not just show everything all at once. Because if you show everything all at once, it will be very difficult to understand. So you need to layer information, what this means. So this means that this 1.5 kilobyte is shown here in this result. So you need to know how to draw the information and how to indicate what is what. So this one, of course, is not in the original journal article because everything is in black and white. But you can point it out to your audience with different colors possible because they mean different things. So this is wild type. All the wild type is shown in blue. All the mutant is shown in red. Okay, so you need to separate your information so that it is directly available to your audience. Here again, we can directly tell them. Okay, wild type production is here, and mutant, there's no production. Because as I told you just now, instead of just only presenting the data, you need to tell them what the data is. Okay, so you need to tell them where to look at and what to look at. So if you remember the second presentation just now, there are many information, many results. So that is a very first layer of presentation only, because she only tells you, okay, I did this and this is the data, right? But she did not explain more or tell you what the data means. So it is our job as a presenter to tell what the data means and why it is useful. So perhaps this is the conclusion for this. Okay, and there are two conclusions according to the paper, for example. So you only need a simplified conclusion and summary, because at the end of the presentation, it must be meaningful. Okay. So we will need to add additional information. However, that is only as a backup. So when you present, it will be good to have some additional information, which is kind of like an appendix. So, for example, you have 20 minutes to present your progress of your research, right? Your project, and you have more data cannot fit into the 20 minutes. Some of it may be supporting data. So this supporting data may be asked during the question and answers, right? So if you have that supporting data, you can keep it after the end of your slides, so that if anybody asks, then you can show them, okay, I did this. I did not present during the presentation because it is too much, so therefore I put it here. Yeah. Okay, so this is the example from my own PhD, which I will not show you the whole thing. However, I will just show you as an overall. Okay. So there are also several things that you can do with the PowerPoint, which makes your presentation better. So one is the slide view like this. So do you know how to do a slide view like this? Yeah. During your presentation. Okay. So in the slide view like this, then you can show everything. And of course, it must be important to present your slide with numbers so that the reviewer can ask you, okay, slide 15, what this thing is about. So at the end of the presentation, do not just like go out of the presentation, but show the slide view, overall view, so that we can understand what it's all about. Okay. Yeah, so this is the slide view. So if you press Control and you zoom out, Control and your mouse to zoom out, then you can see the slide view. And people can ask questions. So in the full result presentation, you may want to show everything. However, in a Viva presentation, you need to only present your thesis according to your experimental results. So therefore, there are differences when you present in a progress presentation and also during your Viva. So your Viva should match what is written in your thesis. So if your thesis has this structure, then you should not introduce any new information which is not in your thesis, but follow the same structure so that it is here. So you can say that chapter 1, chapter 1 is what, which chapter, what is the summary, and so on. So basically, you are summarizing everything according to the structure of your thesis. Okay. So I'm not going to show you the full details because it will be too much to share. Let's go back to this. Okay. So the takeaway is, this is the slide. We need to prepare presentation that is data-driven, understandable, clear, and kind. Okay, so that our audience will be able to understand what we're saying. So of course, we do not have any questions because I hope it is clear enough. But okay, so not all of you have your laptops or computers with you, right? Okay. I wanted to try some exercise where we will just present something that is made up on the spot. So just now at the beginning, we started with the introduction and some keyword. Okay. But you don't have a laptop, right? No. So how can we do this? Okay, so let's try maybe a few short exercises. Okay. So just now we started with the Ramadan keyword. Okay. And we want to continue with another keyword. Okay. Another keyword that we can use. So who would like to suggest a new keyword? Gentle. Yeah. So a new keyword. What should we talk about? Random word generator. Let's try and see if there's any random word generator. So let's try this random word generator. Number of words, 1. Tower. Which, not interesting. Endorse. Number. Aviation. Picture. No, I think this one is still random, right? Still random. Nobody have any suggestions for the next keyword that we'll use? Okay, let's talk about diseases. So I saw the word diseases there. Diseases. Okay. Okay. So just now, who was the last person to speak? Kau. Okay. But Ra'at did not speak yet, right? So okay, we'll ask Ra'at to speak first because just now he didn't get a chance to speak this morning. So you have so how do we so this game or this exercise is where all of us will have 1 minute. So you'll say something based on the keyword. So the keyword is diseases. Okay. Yeah, anything that comes to your mind. So it'll be for 1 minute. And then after you have finished, you'll call upon the next person to speak. Yeah, any disease. Okay. So at first, you can tell us about, introduce yourself and then where you're from and of course the disease. Any disease. Yes. Yeah, anything that comes to your mind. Like this, what do you think? Okay, all right. Okay, all right, no problem. So let's change it. So it's like that. Okay, so who's next? Would you like to call? Next person. You have to call someone. Okay, so Jinda, you have 1 minute to talk about any disease. Okay. So the disease. Oh, alcohol addiction. Okay, alcohol addiction disease. Drug to make you Next, who do you think should be next? Next person you want to call.
I'm not going to talk about anything. So I'm not sure if I don't know I'm already. Yes, although it's that's how I was feeling. Oh, if I saw you, right? I was like, oh, it's a very modern at the top of this is that from suffering from something that suffering that's something. But in my culture, it's not going to happen. Now, from my very first experience about disease, I used to, so when I was 14 years old, I started this from. So, exactly. So, the guy was,
Oh, time's up. So you have to time yourself. Try to summarize in 1 minute. Yeah. So after this, you can share your story. Sounds very interesting. So it's also good to leave a quick hanger for suspense. So people want to listen more to you.
yeah.
Okay, so who should be our next speaker?
Share something that Western method when we diagnose with this conventional method is like a lot of people to get to talk about yeah the and all those things, but the Western method all of the things that is diagnosed is like okay what is very different from Western method. Western method. Some example, chili food, watermelon, very bitter, like fried food.
And say seafood is not good. Get allergy of the problem in the body. Most is seafood and fried.
Yeah, okay. So that is also a different perspective on diseases from the food that we eat rather than from the bacteria or other factors that cause the disease. Next shall be our oh. So you want to skip? Okay. Okay, so who should you skip to? Okay. Next. Okay, so we will come back to you after this. Right, so I'll reset the timer. Okay, so Pranav. Okay, yeah, thank you. Okay, so I can understand about colorectal cancer. And what was the last? Health information, right? And then should we go back now? Okay, yeah, that's a very interesting point of view. And then we have our online friends, Gao and Sana. So who would like to go first, Sana or Gao?
Hello, doctor.
Yes. Could you hear your friends speaking just now?
Kind of, not that much, totally.
Oh, okay.
It's okay. So I have to talk about diseases in my perspective. I'm Sana Yasin. I'm from Iraq, and I will talk about Iraqi. The disease that is common. Like mostly, it's non-communicable diseases. diseases like cancer, diabetic, and diabetic. So most like people here, when hearing the diseases all about cancer or diabetic, no one is like have information mostly about the infectious diseases. So when the COVID came and information about the infectious disease spread and the people was shocking, like everywhere there's a disease, regardless of the cancer or the diabetic. So yeah,
Okay, thank you for sharing. about-
welcome.
Yeah, okay, so next will be Gal. Gal, are you there?
Yeah.
Okay, yeah, so Gal, you can share something about the keyword diseases.
Uh, this is from bacteria, virus, and fungi, and go through injects through the air or water. We can. 好吧，OK。
Okay, thank you. Okay, so that's fine. So we can practice more and more. Okay, so for the next keyword, I think some of you, when you talk just now, when you give your speech or talk, I think you can notice a pattern. So some of us talk with a title. Okay, I'm going to talk about this, and then we give some examples. So that will be good in a sense that there's a structure you are telling or prompting the listener. I'm going to talk about this topic, and you're giving some examples. Some of it is personal examples, or some of it about general information. However, the last part, I think most of us miss the last part, which is how do we conclude so that there is a takeaway at the end of our 1 minute. Okay, so with the next keyword, let's try this, whereby we have title, topic. Second, some examples. They can be anything. And then the points, right? Can be 1 point, can be 2 points. And what's the takeaway? It's a conclusion. Summarize so that somebody can have something to take away at the end of your 1 minute. Okay. So let's select a more fun keyword, which is Pulau Pinang. Pulau Pinang, okay. Or Penang Island. Okay. So it can be about anything. So who would like to start first? Now maybe we call Sana to start first. Our next keyword is Pulau Pinang.
Yes, doctor.
Okay, you can start.
Talking about Penang Island, for me as international student living in Penang and studying there, is a wonderful state to be there because of its diversity and its natural landscape about Penang. And for me, it's representing all the Malaysia, and it's about peace and. You can exploring different parts of life there, like living peacefully, and you can do adventure and doing your shopping in the international branding and all the malls and all the shopping. It's quite satisfying for me. And so like island, this island for me just like it's my second home. Yes. Thank you.
Thank you.
Yeah, so. And who would you like to call next to talk after you?
Sultana B.
Organize.
The island, Penang. I really like the island Penang. Very beautiful. I don't think the traffic here is much. People always complain about too much traffic here. I'm like, the traffic in India. It's really bad there. There is no traffic here. Yeah, it's a beautiful island. Really like it. I really enjoy going to UNESCO World Heritage site and Penang Hill. Yeah, it was a really good experience. Yeah, other than that, because I don't eat much non-veg, so I can't talk much about food, but overall it was a really good experience.
Okay, thank you.
So,
I happy because they stay out of the airport. Never been to such state that you hide the breast. Days pass, now I like the weather, so I managed it. So Penang, it's too beautiful. I like the nature,
槟
like
城
I
是一个
love
非常
the
美丽的城市，
I like
它这里有
hiking
非常多样的文化和饮食，
into
在这里可以
the
品
fact
尝
that despite
印尼的
that
咖喱饭、中国
and
的
small island, so many
烧腊饭、
island groups,
以及
groups,
南下
and
品
then
种
like so
餐
many places to
甚
see
至
around.
还有
So yeah,
Okay, yeah. Thank you. So we can hear different aspects that you introduced from your personal experience, like traffic, see that you like nature, hiking. Okay. Just now, Sana said that she has, I think the more message was that she
还
really
有
liked
一个
it.
感想
Yeah. Okay, so let's. Rad, you would like to go? Okay, Rad is ready.
yeah. I'm working with the International Organization Environmental. I'm like environmental, I'm working very important environmental.
亲切的环境。
After seeing the Penang is very good natural, it's very good environmental because found more species, the animals for species. I like stay found go with the other friend research about of the some snake, some spider, some anything. I like the Penang natural is very good. Have the some idea about of the climate change effect of the found more time raining. This good idea about, but need work with other people in Penang to keep the effect of the climate change. Keep the little use the plastic, little use the anything. This is very important. Keep the natural area, keep the natural the natural environmental. Yeah.
Thank you for sharing about the environmental changes,
Yeah,
climate change affecting all of us. Who do you want to call next to speak after you?
Deal.
Yeah.
Okay.
Yeah, you can start.
Right. So, okay, I'm gonna share you point of view of how the university is. Okay, to me, I think, okay, not presenting all most of the Dell because of it's personal. I think that Penang is like a little Singapore, like pirated Singapore. the people behavior is more or less a bit saying they are proud of their food, even though their food is not that. Okay. Fair enough, but yeah, and also we've encountered a lot of Penang guys, and I kind of think was a bit still a bit careful. So if you don't know what careful is actually, yes.
Yeah.
Like you're scared to lose on something. So you want to like be like competitive all the time, you know. So that makes me think that Penang is like a little Singapore to me. Yeah, another different fun fact that if you speak Chinese or English, right? So actually you notice that there's a difference between the intonation of Penang people and also people. They have to like have the tone a little bit. But overall, I think we have still very great. But be more branding, you know. One thing I want to make clear is that they are pretty bad.
Okay, next.
Care about what we don't want to say.
Okay. Oh,
Yeah, okay.
I think Leo is attacking me. Because I'm very Penang grown girl, so I'm a Penang girl. And yes, we do have to go in which that we always say, "Aya, Penang one better lah." Penang one better. Yes, it is correct because most of us will act that way when people say that they are is better than Penang. And then for a Penang girl, I do really stay at home a lot. That's all. Like this garden is nice or what not, but it don't really tourist place, you know. I was like, yeah, stay at home lah, so Jan, don't want to go out. Yeah, that's all about Penang. Islanders.
Thank you. That is a steal.
As managed
Don't worry, you can go on. It's not very strict.
因为美丽的韩向自由传播。
transport and unable to discuss that much was like very wasn't I don't know how people times you have something just out.
Okay, thank you. And, yeah. I told before my Shanghai is similar with because he also island with and have a mountain on the center of and Xiamen University also have two contacts on in Xiamen. One is on the island and on the language and Xiamen people can say lots of language, South Korean dialogue and Cantonese, Chinese, English, all that. So they are so similar. Good. All right. These seven I shared a lot similar the cultures, language, and the
So the last one will be Gao. So what is your thought about Penang?
I don't want. I want to talk about Penang. Penang is a beautiful city. With a divided country, culture, and food. We can eat Indian curry rice and Chinese roasted rice, and Malaysia native food. It has beautiful coastline and natural sensory, and also have friendly Chinese town and friends. Okay.
Okay, thank you now. So, what should we talk about next?
Let's share your opinion on it.
My opinion about the land. So, I am also from Penang originally. I had the experience of growing up in the island, and also in the mainland. So when I was small, as a boy, when I went to school, I would take the trishaw, you know the trishaw? It's a tricycle, the tricycle where somebody is pedaling at the back. So nowadays it's a tourist attraction. But when I was young, I would take a trishaw to school every day back. So it's almost like a kind of a car at that time. So somebody will fetch you and they'll pick you up from your house, wait for you after school to send you back. But nowadays, the children or the kids are very lucky because their parents mostly will chauffeur them to the school and back, or they will see school buses or school vans, less of public transport. So that's my experience of New York today.
It's not a tourist attraction, it's a scam. 60 ringgit per ride, what the heck? Yeah.
For how long?
For like taking you into the street, the old street in the downtown.
Yeah. But it's a heritage ride, so.
And then I still after the sun. Yeah, very much.
But they can cover up the shade, this like.
Yeah, also one of it.
So the past, the best part of riding there is when it's raining, and all the kids go crazy, so you will get some rain into the trishaw. Okay. So what will be our next keyword? Maybe Rana can suggest the next keyword. Snakes, we want to talk about snakes.
You talk about the snake?
Yeah, it's up to you. What keyword?
I like this animal.
Okay,
Yeah.
so we will choose the keyword snake, and then Rana will talk about it first. Okay, so the keyword is snake. Okay, everyone.
Yeah.
Okay.
Yeah. I have some idea about of the snake in Malaysia because I'm reading because I'm like this animal. The some animals in Malaysia is very special. It's very different any country. The found the largest snake in Malaysia is a friendly snake, but found in Malaysia is very dangerous snake. This very dangerous snake because the more people dies. Is one important of the snake, the south snake viper, is very dangerous. This snake causes the heart stop or the stop the blood cycle, fail the nervous system. This shocking after the 1 hour beating the causes the die. Any people beating this snake. Yeah.
Okay, yeah. So you like to study the Malaysian snake more?
Yeah. Yeah, I'm searching in internet about the special doctor snake in Malaysia. Found the doctor named Nadia in Clinton.
Oh,
Yeah, is a special study on of the snake poison study in the Australia. Australia is very, very famous study about of the potential snake, the threatening snake. Yeah.
So who would you like to call next to talk about?
Yeah, I'm saying the doctor sent email. I need to tell you I need a discussion with you about
Okay.
some snake reported. Now my topic about of the some organism found in most of the snake. The some snake not poison, but caused die. Why? Because found the some small organism after the beginning caused the problem in patient. Autoimmune disease, the mutation, anything. This can cause the disease or cause the die the patient. Yeah. Here can you multiply swap with the animals with the culture the microorganism differential and diagnosis the microorganisms.
So it's not the poison, but
Yeah.
bacteria, microbes that causes
Microorganism, yes, anything.
in the
Yes.
snake.
Yeah, diagnosis the gene with the effect of the human. Can you make treatment the gene of the diagnosis?
So who's next to speak about the snake? Yeah, yeah.
I don't know about you all, do you know that in Chinese culture we have this 12 zodiac? And snake is one of the zodiacs. So in the 12 zodiac we have a rat, ox, tiger, rabbit, dragon, snake. So snake is the sixth position of the 12 zodiacs. So there's one story about this zodiac is that all of the animals are taught how to participate in the race. So the first to pass the finishing line is the rat, and then the second was the ox, and then the sixth was the snake. And then my dad is actually snake. Zodiac. So yeah, that's all I want to share about snakes because I have no further information.
Okay, thank you for sharing.
Found some problem. The any people found phobia about of the snake. And snake is very dangerous. Some people die. The snake is very, very dangerous. Why? The snake is very important environmental. Yeah, because the cycle of the life, the eating the mouse, eating the any
Small
animal.
animal.
Yeah, yeah, it's very important. Not all snake is very dangerous.
Yeah.
It's found more snake is friendly.
Yes. Okay, usually the snakes will go away if you come near.
Yeah, some snake is not poisonous, but is eating.
Defense.
Yeah.
Okay, so who's next after?
Who want to volunteer?
I think Malaysia found the teams help and people found in street.
Okay. Then now.
So I think everyone must know the famous animal in China is the panda. Most of the time, the pandas look like so lovely and so cute if zoom into the bamboos. But panda is also a bear, so it's a very dangerous animal and eat even
大部分是在动物世界上
so in
青蛙
China,
因为我
the children and read the panda enjoy the join the war, like ride the horse or elephants. So that's also a very dangerous animal.
Oh, so you changed the keyword from snakes to dangerous.
No, not all snake dangerous. Maybe.
And now it's dangerous, guys. Stop taking picture of pandas.
Okay, so who's next after?
我认为说为什么是
Okay, so let's talk about snakes. Okay, I've not done any studies about snakes
我认为
before.
说是危险
But personally,
我认为说是危险也认为
because
是
playing myself also quite a hyper person. So, and one of the the worst experience that during that time going down how this cobra in the middle of the mountain. So that was the time that was very funny because I was still young at that time. So I remember very this funny joke that was told by my grandfather. He told me that if you encounter a snake, right? You have to go down, and you so the snake will chase you. You know why? Because if you go down the new snake, eventually was circle itself and then it fall out. I don't know why I just followed that instruction. So I know just going down. Yeah. And snakes are, I think snakes are very friendly animal. And they are delicious to eat. Yeah, personally, yeah, I have eaten snakes because I studied in Sarawak. So when I visited one of the native house, they gave us the grilled snake. Tastes like chicken. Yeah. And then when I visit my one of the relative in China, also they have
我
this
知道
like this alcohol drink.
南美洲
Like wine,
有种
wine
水
over
果
that
蝾螈
drink of snake. So they put snake into the wine and then the
蝾螈
with the wine. Yeah, and they say that actually that what help you become what help you only like that concept in Chinese. So yeah, I'm not that lovely. I'm really snakes for really for it. Still love snakes.
People scared of things. Snakes scared of you.
So they oh good no lah. They still scary man. Right? But yeah, it's true lah. But people right. Actually one of the preliminary mechanism when counter something. You have only it's not there by
Okay, who's next?
what have the problem? Yeah. We don't talk.
There's no way I have experience anything. And also I don't have much about the snake. The only thing that like they are some of them are very and some are like not poisonous. They are some snakes that are not poisonous. They do not do any harm to us. And also some people they keep snakes as their pet. That's good. And yeah, so that's it.
Thank you. Next will be Sultana.
But yeah, I don't know much about snakes, but I used to see this movie when I was in Anaconda. And there's this so scary movie, and I used to imagine if that level of Anaconda is actually real somewhere and it's eating people. And it's so frightening. Yeah, that one memory. And the other one, when I went with Pushpa and Sana to Penang, it was a bit they were pushing and it was so deep. It felt like they were asking us, "Are you guys doing?" And all of them and we were okay. So yeah, I just have this memory.
Was it green in color?
Yeah, yeah,
Green snake.
yeah.
I think it's quite common. It's not dangerous.
Like, oh,
Doctor, in India, we don't
Is it this thing?
when we visit the snake, all the years,
Some occasion.
all the years, we don't give us.
Oh, in
Get
India.
the snake.
In the world, in India alone.
Yeah,
Wow.
a lot.
When you see cobra, then you
Yeah.
but how?
like that you will go up like that, open up the
Yeah.
what do you call it? Like the mouth.
Yeah.
Yeah.
Yeah. We
Very
can
possible.
program with that.
The king cobra also, it's quite big.
The cobra that is yeah.
Hey, Next, Jeremy.
Some people, some people like income from the money response or the next time response. And the snack, the price of snack is very expensive. So, like a snack. So, the things of course. So, so, the better possible snack. So, I don't know.
Is it legal to trade snake? Is it legal to trade snakes, right? Legal? So you can sell snake for only
Certain times.
just being kind of snake. Most of the species are legal. So it's like buying fish and chicken. Hotel is safe.
What you say is correct. If you need to identify the snake, because different snakes have different venoms, right? So for the antitoxin, anti-venom, you need to know what snake has bitten you.
300 types of the venom, even snake.
No,
Right, yeah, yeah. One guy is a physician, but found the antivenom because any types of the special poison. Yeah. Need anything, need the poison.
Yeah.
But I don't know what else they necessarily. But I'm not sure about snakes.
The trade.
Okay, Sana or Gao.
Yes, doctor.
Yeah, so the keyword
Yeah.
was
Okay. For me, I am that one like scared of snake. Even I don't want to know or hear anything about snake. I just keep myself away of snake totally since I was a child. And the only obstacle that I have in Malaysia, it's snake thing. And right now, I'm just thinking about when I will come back, how to deal with the snakes, because they found it around my hostel. One of the thing, and secondly, I haven't seen snake in my life. Only one time that I was with Sultana and Pushpa in the Penang Hill. So this was my first time to saw snakes on in front of me. So I scared a lot, and even I'm not trying to learn anything about snakes thing. I just have total phobia of it. Thank you so much for this not interesting topic. Please just stop the snake thing at all, please.
so the last
Thank
one.
you. Thank you.
You. So Gao, your turn. Okay.
I think I know a little about snake. Most of them are seen in the animal world. For example, they eat frogs. Because in my hometown there is a pool and there is many frogs. As a child, I want to see the snake eat them. Yeah. As a child, I like any animal in the world. I know a snake in South animal called the salamander. Yes, thank you.
Okay, thank you, Gao. So I heard that his hometown has many frogs.
Right. Say that you know there's a Chinese that say that if you're afraid of anything that you should eat it. Yeah, that was what I told when I was a children. My father was afraid of the crab. Then maybe.
Afraid of tiger, it's tiger.
So Sana, we can suggest the last keyword for today because you said no more snakes. So what's the best keyword?
I think one of the topic that I'm interested in, like, it's communication.
Communication.
Yes, how we are dealing with the communication together with the people. Like, I can start like my perspective and my talk about the communication. The simplest thing and the important thing about being with a human, it's how to communicate with people, how to reach the thing that you have to the people in front of you, just like your student or anyone you are presenting your presentation. So there will be some skills you have to learn about the communication. They call it soft skill. It's one of the soft skills. So some of the people, they have that skills and talent from their parents, from themselves, but most of us, like, we have it's learnable skills we have to learn, and it's make our success in our lives and in career 80%. Only 20% is all about the degree and the scientific thing that we are doing and in school and in university, but the 80% is all about the communication and how you build the networking by your communication skills. So I think the communication, it's coming first in all kind of the interpersonal skills that people trying to develop. Thank you so much.
Yeah, thank you, Sana, for the interesting topic. Who would you like to call next? So,
I will go with Yin.
okay.
So I do think that communication skills is very important, especially during interview, especially during job interview. So I personally found that even if you are good at your job, if your communication skills suck, you would most probably won't get the offer to get the job or even get a higher position, because I think communication skills is really that important, because some of the people who have very good communication skills with not really good job handling, they will still get a higher position than you your in your job. Yeah, that's what I found communication skills important in working life. Yeah.
Oh, okay. Thank you. Yeah, so it's really true that sometimes people can communicate really well. They can get a better job or get paid more compared to people who are maybe even more hardworking.
We call that blow work.
Because they can blow one time.
It means that you can really talk a lot, but without doing it.
Who's next after Eden?
Well, communicate. Okay, so I think as well, I am not a person of very to communicate. Okay, but how to my inner mind actually, when I try to communicate, actually always afraid that I will say the wrong things all the time. You know, as I grew up, so yeah, so when I was in my elementary school, I was trying to talk a lot without taking care of those those emotions. So as I grew up, actually, I tend to talk less. I think what I want to say something. So that makes me a lot of anxiety, right? But in terms of like handling messages and information, a lot of it. So, right. Yeah, but in conclusion, that I think communicate is just not art how to practical say. It's also the art of how you take care of things on.
事实上，我认为中国对这方面的教育 或者说至少是
Thank you,
结
Leo.
果当时所处的区域是非常不重视。
Song Chen. Okay.
我在高中的时候才基本上第一次登台演讲。这导致很多
Thank you. So communication is important because you need to make friends and also can learn from each other. Who will be next after you? Sugana.
Like skill. I really don't have any thoughts on communication.
Okay. All right. Thank you. Next.
I consider myself as a person who does not much. And it not many friends. I think since childhood, few numbers of friends till now. So I'm like this because I don't talk much. Is it because I prefer to be like prefer to have this? Okay. So that's I'm also saying through a connection the total people and no. So a way to make connections and all, but again, it is also not that important to have make so many friends and opportunities. I think it satisfies yourself and community. So I would say that it's important for us.
Yeah.
I think it is important to communicate area and I prefer to make a PowerPoint to when I want to share down some knowledge and some ideas to others because I think we enjoy the some strategies here to show you and to show you and not only in job communicating time is very important. I think it's not only about in the job and even in your family. So if you don't have a good communication, you will make a lot of mistakes or something.
Yeah, it's very important to communicate clearly, especially in jobs and things, so that there are no misunderstandings and everybody is looking correctly or the right thing. It's also the same when you have discussions with your supervisors. Ensure that you're communicating clearly and like aligned on the project, understand that we are working on the same thing. Sometimes we say one thing and supervisor may say another thing. Project on the big sometimes So you have to communicate clearer with your supervisors.
Yeah.
Yeah, but if you're not studying it, or you can.
Really?
Next one. Yes, so this now.
Oh, it's relax. Yeah.
Communication is very important project. Communication with me, my work, my job. I work with some place have a special have a special laboratory with the work with the government, work with the environmental, work with the international organization. We do not have any time point between this with a student now. Now lose some work with the environmental with law. Now work with all with the government with the student. With communication now after the student I need more talk with the groups. I need with this work after the student. This publication will be in Malaysia, publication will be a project, English, and food.
Okay, all right. Okay, so it's back to Gao. Oh yeah, there's not Sana again. Okay, Gao. So you can tell us about what you think about communication.
The first thing is that I don't I'm not too good at this. I thought that communication in China, at least me or in my hometown, is very unfocused. The first one, the first times I'm speaking on the stage in my senior high school, and only have 4 times. So that leads a lot of problems. So it's really important. Yes, thank you.
Okay, okay, so that's all. That's all right. So just before we go, it's good to summarize what we have been talking about. So, according to Frederick Sanger, you've heard about him. You know who's Frederick Sanger?
First guy.
DNA sequencing, and also at first he discovered or developed protein sequencing at Peru. And then later on he developed DNA sequencing, so it's very important. And the sequencing, one of the sequencing that we use today is called Sanger sequencing, it's named after him. There is also a Sanger Institute in the UK, also named after him. And he's a very productive scientist. So in his sort of autobiography, which we can read here, it says that there are 3 main activities involved in scientific research. One is thinking, copying, communicating, and doing. Okay, so doing is what we normally do in our lab. This is the base of what we all do. The base means that doing is everything that comes with the work in the lab. So that is the foundation, because by doing research, then you get the data, and from the data, that will lead to new findings or discovery. However, that is just between you and maybe your lab members and supervisor, right? So you discover something, but how do we make that something useful, right? So we have to think about how to interpret or share our findings with others. So it's not only all about being a good scientist in the lab, because if you are just a good scientist, then you are mainly a technician. So technician is somebody whose focus is on the lab, but that's fine. So somebody likes to be technician, they are very good at what they do. Some labs, we have very skilled technicians who work many years and they maybe operate, for example, microscope, scanning electron microscope or some higher end equipment. And then after analyzing our data, we need to communicate this. So there are various ways of communication. Okay, in the past, we will have to type it out on a typewriter. Of course, today we use a laptop and communicate better with visuals and text. Okay, and as an ongoing tradition, we also need to talk about our presentation. So it's not just like having nice slides, but we need to communicate clearly. And communication doesn't mean that we have to have perfect English because all of us come from different backgrounds. And we are, of course, not native English speakers. So we are not expected to have good grammar or to be perfect English, but at least we need to have a minimum understandable standard. Okay, you need to be dark, which is data driven, understandable to everyone, keep it simple. So you have to be clear and to be kind to your audience so that you will be able to have a good and effective presentation or communication. Okay, so with that, we end our class for today and hope that we can practice everything that we've learned today. So I'll share the slides with you later on, so you can lead to the more resources. All right. Okay, so see you again in the next class sometime.
All right.
Bye.
Thank you, Doctor.
So go hiking sometime. Have you gone to the Bukit Jambu?
One of the
One of the trail. Was it to the gazebo or
in the structures.
which one is on the stand one?
Yeah.
Oh. Another one that goes on the left.
Lots of monkeys.
Right? No, sometimes only that monkeys.
Okay, see monkeys all the time.
Oh, yeah, we didn't.
A lot of monkeys. Because even when I drive through the road, a lot of monkeys on the side of the road as well.
Okay, Sana and Gal, that will be the end for today. Bye.
So, now.
Hello，
你们要出去吃饭吗？
Hello，现在去哪里吃？
啊，我不知道，你们去我就去，你们不去我就不去。
2点还有课诶，大哥。
啊，还有吗？
有啊，2点有课啊，网课。
所以他那个新发那个表只是上午的，没有下午的。
有啊。
啊
We have class。自己看有。
我再看一眼。
好。
没有。啊，我自己发微信。 哦，它只是531。哦，我懂了，我懂了，我懂他的意思，就是531。啊？